Hello students. Now in this video I am going to discuss with you regulation of gene expression. Okay, how the gene expression is regulated in the cells. Okay, so first of all let us start with the DNA. Okay, DNA has the coded information for protein synthesis, as you know. Okay, from translation, transcription, and then translation, we came to know all these things. The DNA has the coded information for protein synthesis. These proteins which are required by the cell at different times during the cell cycle. Okay, now the genes present in the DNA, okay, they may not work all the time. All the genes, they do not work all the time, or you can say all the genes, they do not work at the same time. Okay, all together, it's not like that. Okay, so it means that the functioning of these genes which are regulated, okay, by a mechanism, it means that, right? So this mechanism, that we can call it as switched on switched off mechanism. Okay, so whenever the gene is required to undergo the functioning, okay, that is the switched on method, and when it is not required, that you can say is a switched off mechanism. Okay, so this is how the genes they function. They do not work all together at the same time, or they do not work all the time. It means whenever it is required, then. So now there are different types of genes. Okay, the genes, there are genes which are present in the cells, they work, the genes that work all the time. Okay, those genes are known as constitutive genes. Okay, constitutive genes means the genes which work all the time. They produce enzymes all the time. Okay, those genes are known as constitutive genes present in the DNA. Okay, but there are some other genes also present in the cells which work only when it is needed. Okay, then only those cells undergo the process of switched on mechanism. Okay, and they start with the process or the functioning. Okay, so this is how, this is what is called the regulation of gene expression. Okay, the expression, the functioning of the gene is regulated. Okay, whether it will be functioning continuous basis, regular basis, or it will undergo functioning whenever it is required. Okay, so this is what the regulation of gene expression, okay, by the process of, as I said, switched on, okay, switched on and switched off mechanism. Okay, now I can give an example from one of my book. Okay, that how these genes they function. Okay, for example, in the bacteria, that is E. coli bacteria, Escherichia coli bacteria, there is a specific enzyme which is required. Let me rather name the enzyme. It is beta galactosidase, for example. Beta galactosidase enzyme present in the, or which is produced in the E. coli bacteria to break down the lactose. Lactose is a disaccharide. Okay, if you break down lactose, you will get two monosaccharides. Those are glucose and galactose. Okay, so this is the metabolism of lactose. So to break down lactose, this enzyme is required in the E. coli bacteria. So in the cell of this bacteria, when this lactose is present, okay, this galactosidase, this beta galactosidase enzyme is produced. The gene, the specific gene which is responsible for the production of this enzyme is switched on. Okay, and then it is produced and the enzyme further break down the lactose. Now this is in case when the lactose is present in the cell of bacteria, but when it is not present in the cell of bacteria, there is no lactose present in the bacteria, it means there is no need of beta galactosidase to be produced. So at that point of time, that particular gene, okay, is switched off. that undergo a switched off mechanism, because it is no more required to produce this enzyme. Okay, because there is no lactose present in the cell. Understood? So this is how it, they function. Now I'm going to share with you something very, if you understood the basic thing, there are there are many researches, investigations, which have established that regulation of gene activity can occur at any one of the following steps. Okay, I'm going to share with you some of the steps. Okay, and these steps you have to keep in mind. Any one of the following step, okay, the gene expression is, you can say, it is regulated. Okay, what are those? First, transcription level. Number one is transcription level. I'm just going to share with you some of the levels. Okay, at any one of the following levels that I'm going to share with you. Okay, the gene expression is regulated. The gene is regulated. Number one is transcription level. Number two is processing level, or you can say that post transcriptional level. That also we can use this term. Number three is transport of mRNA. Okay, at this level the expression of gene can be controlled. Next is translational level. At this level also the gene expression can be controlled. And then post translational level. Okay, so these are the 5 levels at any point of time. Okay, the gene expression can be regulated or controlled. Okay, these steps that you have to keep in mind. In what happens is that in prokaryotes, if I take an example, like in Escherichia coli bacteria, the gene expression is regulated at the beginning of the transcription level. Okay, so this is how at any step depends. Okay, the expression or the functioning of the gene is regulated. Now if we specifically look into the gene regulation in the prokaryotes, okay, then we have to see in the E. coli bacteria, it is the Jacob and Monod who have discussed about this. Now we are going to discuss and to understand what is Lac Operon. Okay, this is through which we will understand how gene expression is regulated in bacteria. Okay, Lac Operon concept is there in your book. So if you understood this regulation of gene expression, then with this link I am going to start with the Lac Operon. Okay, so in simple word, okay, what is Lac Operon? I am going to draw a sequence of DNA. This is a DNA, for example, present in the bacteria. Okay, prokaryotic organism. It's a sequence of DNA. And what is present in the DNA? The genes are present in the DNA. Okay, so specifically I have mentioned one enzyme here, which is I said that which helps in producing lactose. But now what happens is that in prokaryotes we see 3 different types of enzymes which are actually responsible for breakdown of lactose. Okay, what are those? Number one is definitely beta galactosidase. Okay, so let us write one by one. So one is beta galactosidase. Okay, this is one enzyme. Next enzyme is the lac palmiase. Number 2 is lac palmiase. And number 3 enzyme is transacetylase. Okay, transacetylase. Okay, these are the 3 enzymes and which are required, okay, to break down this lactose in the prokaryotic cell. Now remember that these enzymes are nothing but proteins. Okay, enzymes are nothing but proteins. So in this DNA, specific genes which are required to produce these enzymes. Okay, what are those genes? Here there is a gene present. I am just indicating the genes with the red one. So this is here one gene that is known as I am writing here only Lac Z. Okay, this is a gene which is responsible Lac Y for producing this lac permease enzyme. Okay, and there is Lac A which is responsible for producing this trans-acetylase enzyme. Okay, so specific genes are there which are responsible for this enzymes to produce. Okay, so now apart from this set of genes, we see another set of genes there in the sequence of DNA. What are those? Okay, here this set of gene I am indicating with the green one. Okay, first of all, here we see there is a operator. This is operator. Okay, operator gene. Operator gene. Okay. Operator gene actually what it does, okay, operator gene controls or regulates the functioning of this gene. It regulates the functioning of these genes. Okay, operator gene. Just beside the operator gene, we see another gene known as this is a P, promoter. So this is promoter gene. Okay, promoter gene means here the RNA polymerase enzyme going to bind. Okay, and the process of transcription starts from here. Okay, the RNA polymerase enzyme going to bind here and the process of transcription starts producing mRNA. Okay, apart from this, one more gene is present, that is we can say it is a Lac R. Lac R, it is regulatory gene. Regulatory gene. What it does, this gene, regulatory gene produces repressor substance. Okay, repressor substance. Okay, so these are the gene repressor substance. What it does, that I am going to tell you. So you see, first of all, this is a DNA sequence which is having two sets of gene. One set of gene producing these enzymes, another set of genes that is regulating, operating, and all these sets of genes. So this entire sequence of DNA along with these all genes known as Lac Operon. This is the Lac Operon. Okay, or you can see the mapping of Lac Operon that I have drawn here. Okay, so this is this is technically, this is what we call it as operon. Operon, so sequence of the DNA along with its all the enzymes. Now this what it is actually doing, it is actually intention is objective is to break down the lactose, so we are adding a Lac. So Lac Operon, this sequence of the genes which are actually responsible for metabolism of this lactose present in the cell. Okay, this is what Lac Operon. Okay, now in a two different way this Lac Operon function. Okay, now see, as in the beginning we said, whenever it is required, if lactose is present in the cell of the prokaryote, okay, then it needs to be broken down. If it is not there is no question of breaking down of lactose, right? So if in this lactose is present here, so I am writing first option is in the let's write first of all in the absence, then we will understand. Let us understand this. In the absence of lactose, if there is no lactose present in the prokaryotic cell, is there any need to produce these enzymes? Do you think so? No, there is no need of these enzymes to produce because there is no lactose present in the cell. Okay, so these enzymes are not required. It means, see, these enzymes, as I said, what? These are proteins. Proteins came from where? Proteins came from mRNA. So these genes of the DNA, they first they need to undergo transcription, which will produce mRNA. mRNA will produce these things, enzyme. So as it is not there in the first case, lactose is absent, so there is no need of this transcription process. Okay, so what happens in this case? Okay, this regulatory gene is going to produce first of all one mRNA. Okay, we can name it as a repressor mRNA. Okay, this repressor mRNA which is going to produce a protein known as repressor protein. Okay, this repressor protein is going to bind with the operator site. Operator site. Okay, why operator site? Because operator gene that regulates the functioning of these genes. So we need to stop these genes to undergo transcription. That is why this repressor protein go and binds with this operator site and prevents this gene to undergo the process of transcription. So this is how the further processes are stopped. Okay, this is the first scenario. Second scenario is that number 2, in the presence of lactose. Now if in the cell of the prokaryote lactose is present, now it is required to break down. This is disaccharide, it is to be broken down to get the monosaccharides. Okay, so now these enzymes will be required because they are now going to act on this. So that is why it needs to be produced. So at this time what happens, when this Lac R gene produces this mRNA and the protein, the repressor protein is produced, this lactose goes here as an inducer. It goes and binds with this repressor protein. It goes and binds with the repressor protein. Okay, it inhibits to bind with the operator. So there is no more binding then. So once this lactose goes and bind with the repressor protein, it becomes inactive. Once it gets inactivated, it is not going to bind to the operator site. If it doesn't go to bind with the operator site, automatically what will happen? These genes will undergo the process of transcription. Then they will be producing mRNA. Okay, so mRNA is going to be produced. Okay, and this mRNA is going to produce all these proteins. I have drawn one mRNA here because if you know prokaryotic organism in the cell, we see polycystonic situation. So one mRNA is produced by these genes, that ultimately this mRNA is producing more than one proteins. Okay, the enzymes. So this enzymes are produced and which ultimately will be going and will be acting on this lactose enzyme to break it down into glucose and galactose. Okay, so this is how this Lac Operon function and the genes are regulated. Okay, so you see here they are not the genes are not functioning all the time. Okay, the genes are not functioning all the time. When the lactose is absent, they are not functioning. And how they are being stopped, this by this regulatory gene. Okay, this goes and binds here and stops this. But then when it is present, the lactose is present in the cell, in that case, now they need to undergo the process, further process producing this enzyme, and then it do the breaking down the metabolism of lactose. Okay, so this is how the regulation of gene expression occur in the prokaryotic cell. Okay, so we have done, I have covered two topic. One is the basic concept of regulation of gene expression, and then I entered into the Lac Operon concept, how the gene is regulated, expression is regulated in the prokaryotic cell. Okay, hope it will help you to understand and to produce a quality answer from this explanation. If any questions comes in your mind, note it down and do ask me during my class. Okay, thank you so much for watching the video, and just wait 2-3 more days so that I can be I can upload the next videos. I hope that only 2 topics you are left with, human genome project and the DNA fingerprinting. So I hope 2 more videos, okay, and it will finish, the chapter will finish. Thank you so much. Bye bye.